The human factor. As mentioned before, traditional research on the cerebral cortex of mammals has recognized the quantitative differences, for example, in the period of neurogenesis, number of progenitors, and duration of the cell cycle, that simply relate to the expansion of the forebrain, and particularly the great increase in size of cerebral cortex in primates. Although other unique human characteristics have been neglected, perhaps because so few investigators work on the development of the human cerebrum. In this chapter, we have exposed in some detail those differences, giving some insight on their possible contribution to the specification of the human cortical features. To finish this chapter, we would like to summarize those differences and some others not mentioned before that we think might contribute to establishing the differences in the cognitive potential of the primate and specifically of the human cortex. There is a pronounced gradient of maturation across the large hemispheres in primates that is much less visible in species with smaller forebrains and especially in species with marked altricial growth strategies, altricial, such as mice and rats that exhibit a short, fast brain development.
Altricial.
It means being hatched or born, or having young that are hatched or born in a very immature and helpless condition so as to require care for some time. For example, altricial birds. Okay, the next point. Radial glial cells are primarily prominent in the embryonic primate cerebrum. They start expressing GFAP at the start of corticogenesis, and a subset of them transiently stop dividing. The next point. The primate SVZ subventricular zone is dramatically enlarged and exhibits much more complexity in cellular organization in primates than in other species. Another related feature is the appearance in mid-gestation in humans around 22 weeks and monkey around E72 of a band of tangentially oriented axons that divides the subventricular zone into inner and outer sublayers in some parts of the cortex. Next point, a subpial granular layer has been described in humans that has not been observed in rodents. This is a long-known feature of human development. For example, Brune, 1965, that is, however, systematically ignored at the present time. Next point, the reported differences in the developmental processes between species are reflected in the final cellular output. And, for example, the proportion of GABAergic interneurons exhibits a marked difference between rodents and primates. GABAergic interneurons represent approximately 15% of the total population of neurons in the rat neocortex, whereas in the primates and humans, this proportion reaches 20% in the visual cortex and up to 25% in other cortical areas. In addition, some GABAergic cell types, like the double bouquet cells, are absent in rodents, but can be detected in carnivores and are much more abundant and well-developed in primates and more specifically in human. Double bouquet interneurons are characterized by the presence of a long, narrow, descending bundle of axons that are distributed following a microcolumnar pattern in the neocortex. The axons of double bouquet cells establish hundreds of synapses with a narrow column of tissue, and therefore they have been proposed as key elements in the microcolumnar organization of the cortex, acting on pyramidal cells from different layers within the mini columns. Regarding excitatory that ends the bullet points. Regarding excitatory neurons, differences have been reported in the size and pattern of arborization of projection neurons. Pyramidal neurons from the human prefrontal cortex are bigger than those from other species, have larger and more complex basal dendritic fields, and also have a higher density of dendritic spines. This increase is beyond scaling of cell structure and reflects specific specializations that allow human pyramidal cells from highly associative cortices to receive more synaptic inputs from more diverse origins and therefore to be capable of integrating more information. In addition, specific subtypes have been described, such as the spindle cells that seem to be specific to some mammalian orders, and these are particularly abundant in restricted cortical regions of great apes and especially in humans. These marked differences in the cellular composition of the neocortex from predecessor cells to GABAergic interneurons or glial cells, have to be taken into consideration if we are poised to understand the distinctly human aspects of behavior and cognition and the uniquely human cognitive disorders such as autism, dyslexia, or schizophrenia that probably involve genetic anomalies affecting the regulation of human-specific developmental features.